Welcome to today's session on k-means clustering. So we are going to perform k-means clustering using R programming. Okay. So k-means clustering is one of the clustering methods. So clustering is a unsupervised learning method. Okay, so it is going to group the data based on the clusters available in the current set of data. There is no label for the clusters, so there are no predefined label for the clusters. The clusters are going to be assigned based on the affinity of the data, that is the closeness of the data. Okay, so here we are going to use iris dataset. Okay, so there is a dataset in R programming called as iris dataset. So this is nothing but the data about the flower iris. So it has three varieties: setosa, versicolor and virginica. Okay, so it has set of data which has some data for setosa, some data for versicolor and some data for virginica. Okay, so if you want to see what is there in the iris dataset, you can use head of iris. Okay, so this is the dataset. You can see the columns: sepal length, sepal width, petal width and the species. Okay, so these are the 5 columns available. The species column is the one which defines which flower it is, which variety of flower it is. Okay, so there are totally 150 entries. So 150 entries, we have different set of data for setosa, versicolor and virginica. Okay, so first let me assign this iris dataset to another dataset, iris1. Okay, so with which we are going to build the clusters, k-means clustering. Okay, so I am running this syntax. So iris1 is formed. So from this iris1, I am removing the species column. Okay, the species column is the one which is having the type of the flower, that is setosa or versicolor or virginica. So I am removing that from iris1. Okay, so then I am using the function k-means. I'm using the function k-means, and inside I'm giving the dataset name iris1 and I'm giving 3, the number 3. So that is I'm going to divide the iris1 dataset into 3 clusters. Okay, so and the result will be stored in km. So this is done using the function k-means. Okay, so now let's see what is there in the km. Okay, so km has the different 3 clusters. Okay, the cluster sizes are 33, 21 and 96. That is, there are 33 setosa, 21 versicolor and 96 virginica. Okay, so based on the clustering done. Okay, so what setosa are named as cluster 1 and versicolor is named as cluster 2 and virginica is named as cluster 3. Okay, so this is what we have inside km. Okay, so now we are going to see, we are going to compare what is done using the k-means clustering against the real data. Okay, so the k-means clustering result is stored in km. Okay, the result is stored in km. So we are going to find the cluster of the km. Okay, that is whether it is 1 or 2 or 3. So that I am going to compare against the iris dataset species. Species will be setosa or versicolor or virginica. So and then I am going to form a table out of this to compare. This is to compare whether the predicted is same as the actual. Okay, so what we can see is in the real data set there are 13 plus 17, 50 setosa. Okay, so of which 33 are properly clustered under cluster 1. Okay, and 17 are not properly clustered, so they are clustered under versicolor. Okay, and similarly in the real data set the versicolor count is 50, so of which 46 has been properly clustered into versicolor, that is the cluster 3, and 4 is not properly clustered, so it is clustered as virginica. Okay, so in the real data set 50 are virginica, so all 50 are properly clustered under cluster 3. Okay, so this is the result of k-means clustering. Okay, so let us plot this using a plot. So I'm going to plot iris 1 data set. I'm going to plot the length, the sepal length and sepal width of the iris 1 data set and the clusters which are there in the KM. Okay, so and I'm going to plot the center of the cluster using KM dollar centers against the sepal length and sepal width. The colors of the clusters will be three different colors, so that's why I'm giving 1:3. So and PCH is 8, that is the shape of the plot, plots data points in the plot, and CEX is the font size or the size of the dots in the plot. Okay, so let me run this plot and the points function. So it is plotted, so and the points are plotted. So let me zoom this and see here. So it is plotted against the sepal length and the sepal width of the iris 1 data set. So the red color is showing one cluster, black color is another cluster, and green color is another cluster. So this red color is setosa and black color is virginica and green color is versicolor. Okay, so this shows against sepal width and sepal length. Okay, so this is how we can visualize the clusters formed using k-means clustering method. Okay, so the same thing, the plot can be done using petal length and petal width. Earlier we did with sepal length and sepal width. So the same thing is drawn using petal length and petal width. Okay, and the points are plotted. So let me zoom this and see here. So here also you have three clusters. One is the red cluster, black cluster and the green cluster. So the red one is setosa, black one is versicolor and green one is virginica. Green one is virginica. Okay, so this is how the clusters are actually found using k-means clustering for the real data. Okay, so hope it was a useful session. So we have understood what is there in the iris data set and how to build the k-means clustering using the iris data set by removing the species variable and dividing that into three clusters, and how to analyze the results of the k-means clustering, and how to compare that with the actual data, species and the clusters, and see how it is, what is the accuracy of the clustering. So the clustering results can also be seen using the plot and the points plotted. Okay, so this is done with the two types of plot, one with sepal length and sepal width and another with petal length and petal width. Okay, so hope it was a useful session. Okay, so let's see in the next class.